concerned. Okay, so let's delete that. Actually, I think I'm going to take a screenshot of this first. I don't know why I didn't do that already. Just because it seems like it would be good to have a still image of all of this stuff in addition to a video about it. Come on. Okay, maybe not. Let's try that again. Oh my God, it's driving me nuts. Okay, one more try, and if it doesn't work, I'm just gonna give up. Okay, I guess it's not going to. Yeah, this is not gonna work. Okay, well. I'll just take a still from the video and do that instead, since this is just not cooperating. Okay, so this question was about going over the digestive system cell types, which is great, but caveat, we're not going to be able to go through all of them in this one video, so this is going to probably be a two-parter. And I do want to mention the reason for this approach. I'm going to use my favorite Epic pen color. So I'm also going to include in this list some of their contexts. So we're going to do things like, what is the cell type a part of as far as organs or what have you, and whether it's absorptive or secretory or some other thing. So the reason for this, before I kind of break this down, is something that the person who asked this question and I have discussed as far as ways to approach A&P material, and making sure that you're looking at it from all angles to get the best possible picture of the systems so that you can demonstrate as much mastery as you can, because that's how you earn an A. So one important thing to remember, especially in a situation like A&P where you have multiple levels of organization, so like tissue, organ system, or in the opposite direction, structures within tissues, and then all the way down to cell types and extracellular stuff like proteins and whatnot, is at the end of the day, if you really break it down for any system, cells are the basic building block of life, and cells can specialize. So although they might all have the same nuclear DNA, they do different jobs based on what genes they're expressing. And what that leads to is which genes they're expressing is going to determine their job. And cellular work is accomplished via enzymes, which are proteins. And that is what gene expression functionally is all about for a cell. So cells are going to do stuff with enzymes and express genes for internal housekeeping purposes that are going to be maintaining the cell's own processes that keep it alive. And in addition to that, they're also going to express genes according to what their job is. So if I'm a parietal cell, I'm going to keep myself alive, and I'm also going to engage in enzymatic activity, which allows me to create hydrochloric acid for gastric juice. That's one example. There's many others. But what that means is for an organ system and an organ and the tissues that make up that organ, the work that is being done by that tissue or that organ system, the work that is being done by those things is the sum total of the work that all of the cells are doing. So if we know that the work of an organ system is really boiling down to the work that its cells are doing additively and collectively, then a good way to approach an organ or organ system and be sure that you're kind of capturing all the necessary detail is to think about it from the cells up and be able to describe the cells that are part of that organ or organ system and what they're doing and where they're located. And then you can piece together larger things like homeostatic feedback loops once you've done the cell part. So a lot of students talk to me about like being worried that they're not studying quote unquote the right things, or that they're missing necessary details or connections. And I get that. It's a very real fear and it's an easy thing to do. But if you think about systems from this perspective, where the only work they can do is the work of their cells, it's a good way to make sure you're not missing that specific function and context, because it gives you a baseline or a starting point that you can expand from. So that's the power of going from the cells up, and why I recommend taking the time to do something like this on your own. It shouldn't be your only approach, but it should be part of it. I'm going to say part one, because this is not going to be where we stop by any means. So cells of digestive system, part one, and we're just going to kind of subdivide them into general regions. So we've got let me move this over here. And then we'll do duodenum. So notice I have location in this one part of my little table. And I'm actually not going to do some portions of this because in our class for which this is being generated, we don't spend a whole lot of time emphasizing the oral cavity or its contents. It is part of your reading, of course, but it's not going to be the primary focus for the test, for example, just because we are going to focus more on the major tubular components. So, of course, the mouth is going to include things like the keratinocytes, well, not keratinocytes, the squamous cells that make up the walls of your cheeks and your tongue, and things like taste cells and sustentacular cells, and the cells that make and maintain your teeth, so odontocytes and other things, but we're going to skip those for right now. Those can have their own video later. Similarly, the pharynx is basically a muscular tube lined with stratified squamous epithelium, and it's just a place where food and air and water sort of transit, so we're not going to overly emphasize that. We're going to really start with the esophagus, which is our beginning of our things that do peristalsis and other digestive tube activity. So, as part of the esophagus, we've got squamous cells that are part of the stratified squamous epithelium that lines the esophagus. We want to have an abrasion-resistant surface for potentially rough or abrasive foods to pass by, and create a situation where if a few cells kind of get rubbed off as a food bolus passes, it's no big deal because we've got plenty more. So we've got those. We've also got exocrine cells that are part of esophageal glands. And so we'll talk about what those do in a minute, but basically they're there to produce mucus that lubricates the passage of food. Having a dry esophagus is not ideal. You want stuff to smoothly go through it. And then we've also got fibrocytes that are going to be producing the collagen that's in the submucosa of the esophagus. And as well, fibrocytes that are going to be secreting collagen that is part of the adventitia, because the esophagus is stuck to various other structures nearby and stuck to the body wall using dense irregular connective tissue, which is made by fibrocytes. And then we have smooth muscle cells. And those are, of course, going to make up the walls of the muscularis externa that perform peristalsis. So right now I'm just talking about the different component cell types. So stomach is going to have, in the mucosa, it's going to have columnar epithelium at the top, but those particular columnar cells are not absorptive. They are mucus cells, and they're going to secrete mucus onto the apical surface of our mucosa to protect our gastric mucosa from its own secretions. And then also part of the mucosa are a variety of cells. So we're going to have parietal cells. These are the ones that secrete hydrochloric acid. We've also got zymogenic/chief cells, and these are going to be the ones responsible for creating pepsinogen, which is a zymogen, which means it's an enzyme precursor. So it's not an enzyme yet, but if you do something additional to it, it becomes one. And then we have G cells, which are going to produce gastrin. Remember, gastrin comes from two places. We've got gastric gastrin, and we've also got the gastrin that is secreted downstairs by the duodenum, part of that conversation between the stomach and the duodenum that regulates gastric emptying. And we've also got enteroendocrine cells, which are cells that produce hormones that allow communication between adjacent digestive regions. And in the duodenum, I'm going to skip the smooth muscle cells and the fibrocytes of the stomach for now, because those two things are going to be consistent for the rest of these tubes, because they all have a submucosa and they all have a muscularis externa. So those two cell types are going to function the same in the submucosa and muscularis externa, respectively, in all of the tubes. So there's some stuff that we can just kind of apply globally. So in the duodenum, we are going to have, oops, it should be pink. Enterocytes, these have a lot of jobs. So they're part of a columnar epithelium that lines the entire alimentary canal, including all aspects of the small intestine and the large intestine as well. They have a lot of different jobs that include absorptive function, so absorbing micronutrients, producing brush border enzymes, producing mucus, because some of them are goblet cells. And so they have lots of hats that they wear. So some of them are, well, let's just make goblet cells a separate thing because they're sort of part of the mucosa, but they only produce mucus. So those are going to be alongside enterocytes in the same columnar epithelium, but producing mucus specifically. And then also in the duodenum, we have got the, darn it. So the mucus cells that form the exocrine acini of Brunner's glands. So Brunner's glands are glands that are specific to the duodenum, and they secrete an alkaline bicarbonate-rich mucus, which is going to help protect the duodenal mucosa from the acidic chyme that's coming in from the stomach, and also lubricate the passage of that chyme by virtue of it being mucus in addition to being alkaline. And then that exocrine mucus is alkaline, and it's going to be supplemented by the alkaline fluid secreted by the pancreas from its ductal cells. And all of those things are going to protect the duodenum from the acidic chyme coming in from the stomach, and also buffer it so that the pancreatic enzymes that are going to start to really get chemical digestion going are able to do their jobs, because they only like to function at alkaline pHs. So again, fibrocytes and smooth muscle cells are also going to be components of the duodenum, because the duodenum also has a submucosa and a muscularis externa. And the duodenum is one of those portions of the alimentary canal that is covered on the anterior surface with a serous membrane. So that's going to be consisting of squamous cells and sort of a small lamina propria underneath of loose connective tissue with fat. And then on the back side of the duodenum, there is adventitia, which is dense irregular connective tissue primarily, and that's going to glue the duodenum to the posterior body wall and to the pancreas to help keep it in the proper orientation to the pancreas. So fibrocytes and smooth muscle cells again, because those two kinds are consistent across all aspects of the alimentary canal. So let's see how much time I've got. 30 minutes until I have to be there. Okay. So now what I'm going to do is kind of reduce my pen size and describe the job of each of these cell types. I'm going to have to be fairly brief about it because we don't have that much room. But again, because the approach of this technique is to know and appreciate the diversity of cell types that all must work together to carry off the job of digestion and absorption, and be on top of what their individual contribution to that job is. We don't need to be super detailed here because we're doing an overview. So the time for detail, let's say you are going to zoom in on parietal cells and you're going to closely investigate how they generate hydrochloric acid, which there's a separate video in the short videos playlist for that. The time for doing things like zooming in is going to be after you've done this sort of overview activity, because again, this is providing you with a really robust baseline for how a system gets its work done on the basis of what work all of its cells are doing. And in the digestive system, you can see that the diversity of cell types is considerable. So squamous cells, and I'm going to just casually connect these two things as well, because those are also present. So they're going to be part of stratified epithelium, and they impart abrasion resistance. So anything potentially rough or scratchy, they're going to help mitigate the effects of that. So in the esophagus, there are exocrine cells that are part of seromucus glands in the submucosa. So these are going to be, seromucus means they secrete some serous fluid, which is thin and watery, and some mucus, which is of course gloopy, due to mucin. We all know what mucus is. So seromucus glands to lubricate passage of bolus. Bolus being the term for a compacted mass of chewed up food. Fibrocytes, wherever they are going to secrete collagen / elastin as part of the submucosa, which is the layer that underlies the mucosa all along the alimentary canal. And then smooth muscle cells arranged in orthogonal layers. Orthogonal is just a fancy way of saying at right angles to each other. So this refers to the fact that in the muscularis externa, you've got an internal circular and an outer longitudinal layer, because you need at least two directions of contractile direction to accomplish peristalsis. And then you have a third layer in the stomach to do churning with. So arranged in orthogonal layers to arrange for peristalsis, let's say. Okay, so moving right along. In the stomach, we've got mucus cells. These are also sometimes called mucus neck cells. So I'm just adding that so you're aware of it. The neck portion refers to the fact that at the apical surface of the gastric mucosa, you have this orderly layer of columnar mucus cells, but they also kind of divot in and downward because the neck is the opening to a gastric pit. The gastric pit is what contains the parietal enzymogenic cells as well as the other cell types. But the pit has to have an opening somewhere. So it passes through this neck that's lined with mucus cells at the top. So go look at the histology for more of that. But these are going to be secreting protective mucus to protect the gastric mucosa from the acid and the enzymes that it secretes. Quick spelling note that I have MUCUS here and MUCOUS over here. Mucus is a noun, meaning it's the name for a thing, which is a mixture of water and the protein mucin that forms this gloopy substance that's also lubricating. Mucus, this is an adjective, which means that it doesn't mean anything at all unless I follow it with a noun. So I'm saying mucous cells. So the OUS is the adjectival form. So what we do is if something secretes mucus, we describe it as being mucose. So mucus cells are cells that secrete mucus. So in doing OUS here, I'm communicating that it's an adjective being used to modify the noun cells So I'm not describing mucus, the substance. I'm saying these cells secrete mucus, making them mucose. So I'm belaboring that point because one of the major failings I notice for terminology that students have a hard time with, especially in the context of figuring out which spelling to use for practical answers, for example, is one of two things. One is answering a question with only adjectives and no nouns, which doesn't tell me that you know what you're looking at because you're just describing attributes of a thing that you never name. So I got a lot of answers like dense fibrous for one of the practical questions that was about the fibrous pericardium, but those answers don't get any credit because I'm like, dense fibrous, what? What are you referring to? It would be cool if you told me what you were describing using those adjectives. So seems immaterial, but if you think about medical communication and doing that well and specifically, very important. So if you use an adjective, give me a noun, otherwise no credit. Okay, parietal cells are of course going to be so they secrete protons and chloride ions independently into the lumen of the stomach, which combine with water that comes out of the vasculature in the submucosa to create a mixture of water and those ions, which creates hydrochloric acid that is going to be associating and dissociating according to the rules of those things for acids. So I'm specifying that they're secreted separately because many students assume that the H and the Cl are put together inside the cell and then plopped out as one unit, and they're actually not. They're secreted separately from each other. So there's one channel for chloride and one pump for protons, and they're not actually combined until they're out into the lumen. Zymogenic or chief cells create and secrete pepsinogen, which is a zymogen, and that pepsinogen is turned into pepsin when it hits the acid created by the parietal cells. These guys secrete gastrin, and that's going to be, of course, for chemical communication between the duodenum and the stomach and regulating gastric activity. And then enteroendocrine cells, which create other hormones that are related to digestion. I.e. ghrelin, which is a hormone that's about hunger and satiety. So it helps you tell the difference between being full and being hungry, and it helps tell your brain the state of your stomach. So enterocytes in the duodenum and elsewhere have a bunch of jobs. So absorption of nutrients and water. And they also make brush border enzymes, and those are going to contribute to chemical digestion. Goblet cells. There's that noun form of mucus again, and then the exocrine adjective form, mucose cells of the Brunner's glands secrete bicarbonate-rich mucus, which has the added bonus of being alkaline in addition to being mucus. So it's both buffering and also lubricating, does two jobs. Okay, so that is the starter or part one of cells of the digestive system. And I have to go, so we're going to have to do part two another time. So yeah, I will stop recording and I will